Welcome to the Ross Wonders Meetup. We're very happy to have today as a guest Dr. Alice Shaw, who is now at Dana-Farber. The registration materials included her introduction, so rather than take up time to do that, we will get started. So Dr. Shaw, you have some slides for us.
I do. Thank you so much, Janet. So give me 1 second to pull up my slides. All right, so let me know. Can you see those, Janet?
Yes, thank you.
Great. Okay, well, hi everyone. It's great to see, or to at least know, I heard from Janet and others that we have a number of registrants. I can't see you actually at all on the Zoom, but great to know that you're joining us tonight. Um, and so today I was asked to review the latest data on our newest ROS1 targeted therapy, zidesamtinib. Also, it's called NVL-520, and the data comes from the ARROS-1 trial. There've actually been a number of advances in the ROS1 space the last few years, but I think zidesamtinib really is probably one of the most important ones to know about. And so I'm really excited that we have time tonight to walk through the data together. All right, so here are my disclosures. I've put in bold the most relevant ones. And just to note, I have been working with the company that discovered and is developing zidesamtinib, that's Nuvalent. I started working with them before their inception, and I'm currently a member of Nuvalent's Scientific Advisory Board. All right, so I thought I would start with a brief background on ROS1 positive lung cancer before diving into zidesamtinib. So shown here is a pie chart that summarizes the known oncogenic drivers in lung adenocarcinoma and their frequencies. Now, just for definitions, oncogenic driver refers to an oncogene or cancer-causing gene that's been activated by a mutation or a chromosomal rearrangement, and that's driving the uncontrolled growth and survival of the cancer. And as a result, these cancers now become dependent on or addicted to the oncogenic driver. And so blocking or inhibiting the oncogenic driver can lead to cancer cell death and really dramatic responses in the clinic. And so here you can see that there are many different oncogenic drivers, but I think for everyone watching tonight, the most important one, of course, is ROS1, which, as you probably know, is relatively rare, seen in about 1 to 2% of lung adenocarcinomas. But I would say that it's an incredibly important target to identify in patients because of all of the highly effective targeted therapies that are now available. And so I wanted to spend a few minutes on the ROS1 targeted therapies that have been developed over the last 15 years or so that we can really understand the scientific and clinical context for developing zidesamtinib. And so the first ROS1 inhibitor that we developed was crizotinib, and we often refer to crizotinib as the first generation ROS1 inhibitor, or TKI for tyrosine kinase inhibitor. Now, crizotinib, as some of you may know, is also an ALK inhibitor, also a MET inhibitor. So it's really a multi-targeted TKI. And I think many of you know that crizotinib is important because it really launched the whole field. Early on, we noted that ROS1 patients can respond sort of extraordinarily well to crizotinib. This is one of my patients, I think one of the very first ROS1 patients ever treated with crizotinib. And you can see the dramatic improvement in his disease after just a few months of crizotinib treatment. Based on the data we generated from the phase 1 study, crizotinib was for a number of years the standard first-line therapy for patients with advanced ROS1 positive lung cancer. In fact, I still have a number of ROS1 patients on crizotinib now going on over 10 years. So it can be very, very effective for some patients. Soon after crizotinib was in the clinic, we started testing another ROS1 inhibitor, this one entrectinib, because it was more potent and also had better brain penetration than crizotinib. And soon, entrectinib became another standard first-line option for patients with advanced ROS1 positive lung cancer. But in the clinic, we started to see patients relapsing on both crizotinib and entrectinib. And we then spent years studying why patients relapse, in the clinic as well as in the lab. And so for both crizotinib and entrectinib, we identified resistance mutations within ROS1 itself that cancers develop to try to evade these drugs, with the most common resistance mutation in ROS1 called G2032R, and we often call that a solvent front mutation. But in addition to resistance mutations, we also noticed that patients, especially on crizotinib, frequently relapsed in the brain due to crizotinib's known poor penetration of the blood-brain barrier. And so several companies set out to address these liabilities of the early generation ROS1 TKIs, and they developed new next generation ROS1 TKIs, so repotrectinib and taletrectinib, both of which can overcome that G2032R resistance mutation, and both of which also penetrate into the CNS. And so these TKIs were initially tested and developed in patients who had received prior crizotinib, but eventually they moved into the first line setting, and they've now basically replaced crizotinib and entrectinib as the preferred first line therapies for advanced ROS1 positive lung cancer. But both repotrectinib and taletrectinib also have their limitations. And these limitations, which I'll talk about in the next two slides, really have driven the rational design and development of yet another next generation ROS1 TKI, and that is zidesamtinib. Zidesamtinib, it's a mouthful, for patients who've received prior ROS1 inhibitors. And so on this slide, I've summarized the efficacy seen with the earlier ROS1 inhibitors, so crizotinib, entrectinib, repotrectinib, and taletrectinib, when these TKIs have been tested in newly diagnosed patients. So this is the first line setting. And I've also shown here the efficacy that we typically see with standard platinum-based chemotherapy given in the first line setting. Now, these data, of course, are taken from separate clinical trials, and there are, of course, limitations in this type of cross-trial comparison. However, just to highlight that, maybe not surprisingly, the targeted therapies are all much more effective than chemotherapy in terms of response rate and progression-free survival, which is a measure, of how long the drugs keep the cancer from getting worse. And you can see that the response rates with ROS1 TKIs range from about 70% to 90%. So the vast majority of ROS1 patients do have a reduction in tumor burden when they're treated with the targeted therapy. And on the right, you can see here progression-free survival, and you can see actually that the newer ROS1 targeted therapies that were developed to overcome resistance mutations and to penetrate the brain, so this is repotrectinib and taletrectinib, we do see longer progression-free survivals than the earlier generation TKIs, crizotinib and entrectinib. So improving potency, improving the coverage of resistance mutations, improving brain penetration, all of that does seem to translate into improved efficacy with much longer responses in the clinic. Oops. But I did actually want to emphasize 3 points about the efficacy. So first, I want to say that these data that I'm showing you here are in newly diagnosed TKI naive patients. And in patients who actually have received a prior ROS1 TKI, such as crizotinib, the efficacy of repotrectinib and taletrectinib is notably less. So the response rate is more like 40 to 50%, and median progression-free survival is more like 9 or 10 months. The second point that I wanted to make is that while first-line repotrectinib and taletrectinib can lead to very durable responses, as you see here, patients still do eventually relapse due to resistance. And so we do need treatment options for patients who've received prior repotrectinib or taletrectinib. And then the last point I'll make is that I do think we can do even better, certainly in previously treated patients, but I would say also in this first line setting. And some of you may know that in ALK positive lung cancer, which is a related but distinct type of lung cancer, we have a third generation targeted therapy, lorlatinib, that induces really remarkably prolonged responses in the first line setting. Likely the median progression-free survival with first-line lorlatinib in ALK-positive lung cancer is going to be beyond 7 or 8 years. And so this is what I hope we could achieve with an even better ROS1 inhibitor. So the other major reason that I think we need a better ROS1 inhibitor is because of the side effects of the currently available ROS1 TKIs. And I would say that for me, this is really the most important limitation of the newer ROS1 TKIs. And these are important because, they can really impact a patient's ability to maintain efficacious dosing, and they can definitely impair a patient's function and quality of life, especially if we're giving these drugs over a long period of time. And so here I've summarized just a few key aspects of the safety profiles of entrectinib, repotrectinib, and taletrectinib. There's a lot of data on this slide, but I just wanted to highlight a few points around safety and tolerability. And so focusing on repotrectinib and taletrectinib, you can see that just over one half of patients experience severe, grade 3 or higher side effects during the course of treatment. And, about 37-38% of patients will actually require a dose reduction. Fortunately, though, only a small percentage of patients, 2% up to 7% or so, need to completely discontinue the TKI due to treatment-related side effects. And so what are these side effects? Well, you can see here that repotrectinib actually can cause a variety of different side effects. But I would say that in my experience, the most common and troublesome side effects are dizziness, dysgeusia, which is a change in taste, and neuropathy or paresthesias, which is like pins and needles or numbness. And these side effects are likely related to repotrectinib inhibiting another target called TRAC, and in particular TRAC-B, which is related to ROS1. In fact, with entrectinib, which also inhibits both ROS1 and TRAC, you can see that entrectinib also causes similar TRAC-related side effects, so dysgeusia, dizziness, and paresthesias. Now, with taletrectinib, there can be some TRAC-related side effects, but these are much less common. And instead, with taletrectinib, there's more GI toxicity, including liver toxicity, as well as nausea, vomiting, diarrhea. Although in my experience, these GI side effects are usually mild and manageable. But in any case, I think overall, these safety data do highlight the need for a ROS1 inhibitor that has enhanced selectivity for ROS1 with fewer off targets such as TRAC so that we can improve on safety and tolerability. So this brings me now to zidesamtinib, which, as I mentioned earlier, was designed and developed with all of these limitations in mind. And so maybe before walking through the clinical data, what we'll do is talk about the preclinical data, which really does capture key features of this molecule, and I think really helps explain the promising data that we're seeing in the clinic. And so first, zidesamtinib was rationally designed to be incredibly potent against ROS1 and known ROS1 resistance mutations, such as that solvent front mutation G2032R. So here on the left, you can see that based on cell line data in vitro, zidesamtinib is more potent against wild type or non-mutated ROS1 and the most common ROS1 resistance mutations compared to these other ROS1 TKIs, including repotrectinib and taletrectinib. On the right is what we sometimes refer to as a heat map. And this heat map allows us to quickly visualize the differences in potency of different ROS1 inhibitors. So you can see that each column represents a different ROS1 inhibitor with zidesamtinib on the far right. And then each row represents a cell line model with a ROS1 fusion, and in some cases, a ROS1 mutation as well. And the green color on the heat map indicates greater potency, while the red color indicates no potency and yellow is intermediate. And so I think what you can appreciate here is that zidesamtinib is highly potent against all of the different ROS1 fusions tested and maintains that potency even in the presence of a ROS1 resistance mutation, in contrast to other ROS1 inhibitors that lose potency in the presence of a ROS1 resistance mutation, especially the G2032R mutation. So zidesamtinib is highly potent against ROS1 and ROS1 resistance mutations. The second key feature of zidesamtinib, which was very intentional on the part of Nuvalent, is its enhanced selectivity for ROS1. And so on the left is what we call a kinome selectivity tree, where we're depicting at a high level all of the different kinases and how closely related they may be, and then using circles or dots on the tree to indicate which kinases are inhibited by zidesamtinib. So here you can see that zidesamtinib inhibits ROS1 most potently, followed by ALK. It's in fact a very potent ALK inhibitor. And then you can see that there are only a few other kinases that are modestly inhibited, about 10 to 50-fold less so than ROS, though. And that includes that problematic off-target called TrkB. And this is further highlighted over here on the right. Showing how much more selective zidesamtinib is for ROS1 over TrkB compared to other ROS1 TKIs. And in fact, you can see here with repotrectinib that, in fact, repotrectinib is a much more potent TRK inhibitor than ROS1 inhibitor. So there's much greater selectivity with zidesamtinib, which we predict should lead to fewer side effects, especially those that are TRK related. And then the third key feature that I wanted to highlight, and again, one that Nuvalent was very focused on, is this ability of zidesamtinib to penetrate the brain. So, as many of you know, this is a really important feature of our targeted therapies because we need a drug that can both treat existing brain metastases, but also one that can prevent the development of brain metastases. And so here you can see on the left that in a preclinical animal model, zidesamtinib can penetrate into the brain to a similar extent as lorlatinib, which does have proven CNS penetrance and activity in the clinic. Both zidesamtinib and lorlatinib actually appear to have much greater CNS penetrance than other ROS1 inhibitors, such as repotrectinib and taletrectinib. And then on the right, you can see that in preclinical models in vivo, zidesamtinib has significant anti-tumor activity, inducing much deeper and more durable responses, intracranial responses, than either repotrectinib or taletrectinib. So potent CNS activity with zidesamtinib, and that really should help to treat and prevent brain metastases in patients. So this now brings us to the ARROS-1 clinical trial. So this is an ongoing global phase 1/2 clinical trial of zidesamtinib in ROS1 positive lung cancer and other ROS1 positive tumors. The phase 1 portion was a typical dose escalation where we tested escalating doses of zidesamtinib from 25 to 150 milligrams daily in patients who had received prior ROS1 TKIs. So over 50 ROS1 patients were treated in dose escalation, and this allowed us to establish the recommended phase 2 dose of zidesamtinib, which is 100 milligrams daily. Now, in the phase 2 portion of the study, we enrolled patients into specific expansion cohorts based on prior ROS1 TKI exposure. So you can see that we enrolled patients who were TKI naive. In fact, we're still enrolling ROS1 TKI naive patients in the study. We've also enrolled patients who received one prior ROS1 TKI and patients who received 2 or more prior ROS1 TKIs. And as of last March 2025, the study had enrolled a total of 514 ROS1 patients across the phase 1 and phase 2 portions, including lung cancers and other cancer types. So the safety population, which I'm going to show you today, includes 432 patients with ROS1 positive lung cancer, all of whom received the standard dose of 100 milligrams daily zidesamtinib. And then among these 432 patients, 117 define what we're calling the pivotal efficacy population, which consists of ROS1 TKI pretreated patients with at least 6 months of follow-up. I'm also going to share some emerging data from 35 ROS1 TKI naive patients who had been treated by August 31st, 2024, so that we have a reasonable duration of follow-up on these patients. And so focusing on the TKI pre-treated patients, shown here are the clinical characteristics of the 117 patients who had received one or more prior ROS1 TKIs. The clinical characteristics that you see here are really consistent, with our original report describing ROS1 positive lung cancer back in 2012. Here you can see that the average age in this cohort was 57 years old. That's, of course, younger than the average age of typical lung cancer patients. Almost 70% of patients here are never smokers. Of course, we know that ROS1 is not a smoking-related lung cancer. And then here at the bottom left, you can see that half of patients had brain metastases, and just over 1/3 of patients actually had known or documented ROS1 resistance mutations, which is in line with what we've previously reported on in terms of the frequency of ROS1 on target resistance after prior ROS1 TKIs. Here on the right side of the table, what I wanted to highlight are the prior ROS1 TKIs that patients had received. You can see that about half of patients had received one prior ROS1 TKI, while a half of patients had received two or more TKI. So this really is a very heavily pretreated population, and more heavily pretreated than the patients who were on the trials of repotrectinib or taletrectinib. For patients who received one prior ROS1 TKI, it was pretty evenly split here between crizotinib and entrectinib as the prior ROS1 TKI. And then in patients who had received two or more ROS1 TKIs, you can see that about 3/4 of patients had received prior lorlatinib, which does have ROS1 activity, and 1/4 of patients had received prior repotrectinib, and just a smaller number had received prior taletrectinib. So shown here is the efficacy of zidesamtinib in this previously treated population of ROS1 positive lung cancer patients. So across this entire group, the objective response rate was 44%. And then focusing in on those ROS1 patients who had received just one prior ROS1 TKI, you can see the response rate was 51%. Now, for comparison, in separate trials, we've seen that both repotrectinib and taletrectinib also do have activity in the setting of one prior TKI, though for the most part, those patients had received crizotinib as their prior TKI, whereas here with zidesamtinib, we have about an even split of crizotinib and entrectinib. But I would say this response rate of 51% does seem to be at least on par, if not better, than what's been reported with repotrectinib and taletrectinib. In the waterfall plot shown here, just to highlight that the majority of patients do have some amount of tumor regression, even if they don't meet formal criteria for a response, and also to highlight that patients who've received a prior ROS1 TKI and chemotherapy, those are highlighted by these hatched bars, also really respond well to zidesamtinib. I also wanted to highlight here that zidesamtinib does have efficacy after two or more ROS1 TKIs. And so shown here, the response rate among this subpopulation of patients is 38%. And while the numbers still are small, there does appear to be very promising activity of zidesamtinib even after prior repotrectinib or taletrectinib. With prior repotrectinib, the response rate was almost 50%. Shown here is the data on duration of response, or DOR, in patients who received prior ROS1 TKIs. And again, focusing on those patients who've received one prior ROS1 TKI, you can see that almost all patients, so 93%, remained in response even after 18 months. And then focusing in further on the 12-month DOR rate, you can see that compared to 93% with zidesamtinib, repotrectinib and taletrectinib have a 12-month DOR rate of about 60%, suggesting that zidesamtinib may be able to induce more durable responses than either repotrectinib or taletrectinib. What about progression-free survival in these TKI pretreated patients? So progression-free survival captures all the patients, not just the ones who've achieved a response. And here you can see that about 70% of patients remain free of progression even after 18 months of treatment, which is very impressive, especially given that median progression-free survival with repotrectinib and taletrectinib is only about 9 to 10 months after prior crizotinib. And shown here are additional and important aspects of zidesamtinib's efficacy in previously treated patients. On the left is efficacy in patients who've received any prior ROS1 TKI and have a documented G2032R resistance mutation. And consistent with the preclinical data I showed you, you can see that zidesamtinib can induce responses in a significant fraction of these patients, and these responses do appear really durable. On the right is the efficacy of zidesamtinib in patients with measurable brain metastases. Here, the intracranial response rate was 48% in patients who had received any prior ROS1 TKIs and 85% in patients who had received prior crizotinib as their ROS1 TKI. You can even see that there were complete responses, so CRs that some patients achieved. And again, the durability of these responses was really impressive. Over 90% of the CNS responses here in crizotinib pretreated patients were ongoing beyond 12 months. And that's really consistent with the CNS penetrance and the intracranial activity noted in preclinical studies. Before moving to the safety profile, I did want to spend just a minute on the emerging data with zidesamtinib in TKI naive patients. And so here you can see that among 35 TKI naive patients, the response rate was 89%. And here in the waterfall plot below, you can see that every patient achieved some amount of tumor regression, with most achieving a partial or even complete response. Now, the data are still maturing, but so far these responses appear to be durable, with 96% of patients who had achieved a response still responding at 12 months or beyond. On the right is the emerging CNS data with zidesamtinib in TKI naive patients. Here, the numbers are still quite small, but certainly consistent with what we've seen in the previously treated setting, with almost all of the patients responding significantly in the brain, including 4 of the 6 patients having complete intracranial responses. So very, very promising data. And as I mentioned, we are continuing to enroll TKI naive patients to the ARROS-1 trial so that we can build a even more robust data set. Now, in terms of safety, I think this is where zidesamtinib, in my experience, has really clearly differentiated itself from other ROS1 inhibitors. So shown here is the safety data with zidesamtinib from the 432 patients in the safety population. These are all patients with ROS1 positive lung cancer who received the standard 100 milligram daily dose. Here on the left is a table that summarizes treatment emergent adverse events. So this is TEAEs. So these are any side effects that were reported after a patient started treatment, regardless of whether they were attributed to the drug or not. Now, this table just shows TEAEs that were reported in 15% or more of patients. So this is not a complete listing of side effects, just a summary of the most common ones. And here you can see that the most common side effects are peripheral edema or swelling, constipation, increased blood levels of CPK, which can indicate, irritation or injury to muscle cells, fatigue, and dyspnea, which is shortness of breath. I think what's notable here is that these side effects even are not that common, and they're actually almost all mild rather than severe. And I think what's also notable is that we're really not seeing a lot of side effects related to TRK inhibition. So we're not seeing that almost any dizziness or paresthesias, which, as I mentioned, can make repotrectinib very difficult to tolerate. Also, other than constipation, we're also not seeing a lot of GI side effects, such as nausea, vomiting, or diarrhea, or liver toxicity, which, as I mentioned, we do see with taletrectinib. Here on the right, you can see that only 10% of patients treated with zidesamtinib have required a dose reduction. You may remember that I mentioned that over 1/3 of patients treated with repotrectinib or taletrectinib do need to dose reduce due to side effects. And then consistent with its favorable side effect profile, there's really only a 2% rate of discontinuation of zidesamtinib due to side effects. So to summarize, I think the ARROS-1 data with zidesamtinib does look incredibly promising. I think it shows that through iterative learnings and rational design, we can really optimize our targeted therapies and likely further improve on outcomes for patients. And I would say for zidesamtinib in particular, we are seeing very durable activity, including in patients who've received one or more prior ROS1 TKIs, and that includes prior repotrectinib and taletrectinib. I think zidesamtinib also appears to be especially active in patients who have evidence of the G2032R resistance mutation and other mutations, and that's, of course, in line with the design of the molecule and all of the preclinical studies. Zidesamtinib also shows potent intracranial activity, including in patients who've received prior CNS penetrant TKIs, such as entrectinib, repotrectinib, lorlatinib, taletrectinib. And I mentioned before, I think this is a really critical feature of next generation targeted therapy so that we can both treat and prevent CNS metastases. And then lastly, I would say the safety profile of zidesamtinib does look extremely favorable. The side effects are mostly mild and manageable, and we're really not seeing much in terms of track-related side effects that have really kind of plagued other ROS1 targeted therapies. And so based on all of the data, how does zidesamtinib fit into the therapeutic landscape for ROS1 positive lung cancer? This is a slide that I showed earlier, really showing some of the typical treatment approaches that are being taken for patients who have advanced ROS1 positive lung cancer. And here's how I think that zidesamtinib in the short term will sort of fit into this landscape, which is really after a prior ROS1 inhibitor or prior sequential ROS1 inhibitors. But I would say that, based on the improved potency and CNS penetration, and also the ability of zidesamtinib to prevent resistance, I do think that where zidesamtinib may ultimately land is in the first line setting, because we have seen kind of time and time again in other oncogene-driven lung cancers that using our best targeted therapy up front maximizes the benefit to patients. So this is my final slide, and I just wanted to, comment that I think even beyond improving on efficacy and safety, which already is huge, I do think that zidesamtinib also offers the opportunity to build combinations and multimodal regimens so that we're not just controlling disease over the long term, but hopefully eradicating disease to try for cure. And so in the current treatment paradigm, when we use sequential ROS1 targeted therapies, the challenge that we face is the evolution of increasingly heterogeneous and complex resistance mechanisms. And this can prove really difficult for us to treat effectively in the clinic. And I would say that instead of this approach, I think we'd much rather start with our most effective targeted therapy, perhaps as the anchor in a combination approach, then go after any residual disease with local therapy, such as radiation or even surgery, then followed by maintenance with that anchor TKI to really ensure that, any stray cancer cells are indeed eliminated. This is a very kind of futuristic, thinking, but I do think zidesamtinib now allows us to really plan for the future and to plan for future treatment paradigms that can really push the boundaries of what we achieve even in the advanced setting. So with that, I am going to pause here and thank you all so much. I wish I could see you, but I can't, but I'm certainly happy to take any questions.
Thank you, Dr. Shah. That was a lot of information. If people in the audience have any questions, go down to the bottom of your Zoom screen. There should be a Q&A button. If you don't see it, click on the more that has three little dots and find the Q&A button, and you can submit your questions through Q&A, and I'll pass them on to Dr. Shah. We already have one, but I want to catch something before we go to that. So if somebody who has ROS1 positive cancer has brain metastases, would you recommend that they try a TKI first before treating with radiation?
So thank you, Janet, for that question. It's actually not an uncommon question that does come up because, of course, we do, through the course of, I would say, a number of years treating patients, but also in the literature, we probably do see 20, 30% or more of patients with ROS1 positive lung cancer who do present with brain metastases. So this is certainly not a rare occurrence. I would say, Janet, it really depends on the situation. but a common situation is that a patient undergoes, at the time of diagnosis, a brain MRI for staging. They're actually don't have any symptoms, and the brain MRI picks up some small, incidentally identified brain metastases. Might be one little one, or might be multiple little ones that aren't causing any issues. And I would say in that situation, which in my experience is the most kind of common situation when brain metastases are discovered, I would say I would definitely not pull the trigger on radiation. I would absolutely consider starting a CNS penetrant ROS1 TKI because we have so much data now showing that the CNS penetrant ROS1 TKIs are very, very active in the brain. And as I mentioned, can very effectively treat brain metastases and going forward, prevent further development of brain metastases. So I would say that yes, in general, oftentimes we can choose TKI over radiation. There are exceptions, though, Janet, and just to mention. So, for example, if a patient has a very large, symptomatic, large brain metastases, that's going to cause an acute neurologic, compromise. Those are different situations where we may even have to consider neurosurgery first to alleviate the brain metastasis acutely.
Thank you for that. Wow, we just had a whole bunch of questions pop up. I don't think we'll have trouble keeping busy.
No, oh
Uh,
my goodness, I see them too now.
so one that's come up in a couple of places. When do we estimate that zidesamtinib might be available for use in the US off of study? And I believe the date is set in September of 2026 to be considered. Is that correct?
Yes. So the data that I shared with you today is data that will support, we hope, the initial approval of zidesamtinib in the U.S. And as you said, Janet, we've heard that approval could come in the September 2026 timeframe. But of course, we can't know for certain. And as you know, for the time being, though, until that approval happens, we do have the expanded access program of zidesamtinib open at a number of sites. So we still can access zidesamtinib through the EAP. And I mentioned that the AROS-1 trial, though, is still enrolling TKI naive, so newly diagnosed ROS1 patients as well. So that's another way for patients to access the drug.
Do we have information for people accessing it outside the US or when it might be submitted for approval outside the US?
So I don't have as much data. I don't have as much information around ex-US, Janet. So that's probably a question that we would ask our Nuvalent colleagues.
Thank you. All right. So zidesamtinib seems to be the most effective TKI against the majority of our resistance mechanisms. What happens when it stops working? What do you do for patients?
Yes, so that's an excellent question. And I would say, in general, whenever we have any targeted therapy that's been working for a patient and has stopped working now, what we've seen over the years that it is really important to try to understand why the drug may have stopped working. So to understand if there's a targetable resistance mechanism that underlies the relapse. And so for anyone who's relapsing, we do usually recommend at the minimum a liquid biopsy, so looking at circulating tumor DNA, and if it's possible, safe and technically feasible, even a tumor biopsy as well, because that often can provide information that we can't see in a liquid biopsy. And what we're looking for, as I mentioned, are targetable alterations that may be driving resistance and that we may be able to overcome. So, as an example, I think with zidesamtinib, it's too new. We don't know the resistance mechanisms that we may see. If I had to guess, though, Janet, I would say we may not see on-target ROS1 resistance because zidesamtinib is so effective at overcoming and preventing these ROS1 resistance mutations. So that means that we may see non-ROS1 mechanisms of resistance. And I would say a common one that we could see would be off targets such as MET, for example. So MET amplification is a very well known and well validated mechanism of resistance resistance to a whole host of different targeted therapies, EGFR, ALK inhibitors, ROS1 inhibitors. And so one of the main reasons we do these biopsies is to look for alterations that are potentially targetable, like MET amplification. For MET amplification, those patients can be very responsive to the addition of a MET targeted therapy such as tepotinib or capmatinib. That's been reported in the literature, again, across kind of in general, not specifically to zidesamtinib, but in general, adding a MET TKI can often reinduce sort of responses in patients who have relapsed to the original targeted therapy. But there are other mechanisms of resistance as well that we're just learning about. So I do think continuing to really push on the research and understanding resistance is really critical to then having next steps for our patients.
Thank you for mentioning rebiopsy. So I know that in practice, some people's doctors are simply recommending they try the next TKI without rebiopsy. Is liquid biopsy a good option if they can't get a tissue biopsy?
I do think liquid biopsies, well, first of all, they have gotten better and better over the years. And I think the liquid biopsies can be very informative. But they do have limitations in terms of sensitivity. So there are, of course, limits of detection. And I would also say that liquid biopsies can be particularly challenging in looking at copy number changes. So looking at I mentioned MET amplification. So trying to find MET amplification on a liquid biopsy can be difficult. Also looking for loss of genes on a liquid biopsy can be exceedingly difficult. So for example, looking for loss of the MTAP gene, we really can't do that on a liquid biopsy. So I do think liquid biopsies are a good first step when we're starting to work up a patient who may have developed resistance. But we often do need to go to that next step, which is a biopsy of the resistant tumor itself.
Thank you. A couple of people have asked for additional information about where the trial sites are, and we will share in the Ross Wonders group, again, the link to the trial. All of the expanded access in the US is at trial sites. So we can follow up with that.
Great.
Okay, so one of the things that's come up repeatedly and that I'm not sure we have an answer for is why is immunotherapy not working for a majority of patients? And would you recommend immunotherapy as a treatment after all TKIs have been exhausted?
Yeah. So that is another excellent and very timely question, of course. And we do want to make sure we've exhausted all possible options for patients. So it's natural to wonder about immunotherapy. But there is a lot of data now over the years really showing that patients with these types of rearrangements, ALK and ROS1, really respond poorly to immunotherapy. So checkpoint inhibitors like Keytruda or pembrolizumab or Opdivo, nivolumab. In some series, the response rate has literally been 0% among ROS1 patients. So clearly, just not very responsive. And so we generally tend to not use checkpoint inhibitors, certainly not when we have other established options. Now, the first question is really why? Why are ROS1 patients so much less responsive than other lung cancer patients? And I would say there are a few reasons. One is I mentioned during my talk that we know that ROS1 positive lung cancer is not associated with smoking. It's a never smoking lung cancer. And in the setting of never smokers lung cancers in general, we know that the overall tumor mutation burden is very low. So there just aren't that. We know there's ROS1, of course, and there are some other mutations, but there's not a whole slew of mutations the way a smoker's lung cancer may have a huge amount of genetic alterations. And because of that, then there are fewer what we call neoantigens, these mutated proteins that look foreign to the immune system. And so it's just much harder then for the immune system to recognize a never smoker's lung cancer as foreign and to try to then be able to attack it. The other reason is that we've seen, again, in never smokers lung cancer in general, that there's just not, it's just a very, what we call immune desert kind of cancer, meaning that there're just not many T cells that are infiltrating the cancer. So it's very quiet from that standpoint. So the T cells aren't even around to be able to, again, recognize and fight the cancer. So this is a reason why, we the underlying reason why immunotherapies probably don't have very much activity. Now, having said all of that, I would say that if I have a ROS1 patient who has been on, you know, ROS1 targeted therapies, including the recent ones, have been on chemotherapies, including platinum, pemetrexed chemotherapy. So they've been on therapies that have established activity. They may even have been on investigational trials. If they are still in need of options, we do sometimes consider immunotherapy, Janet. So we might do that, but it's generally in a later line setting. The other thing I'll just say before turning it back to you is that we do not generally like to combine TKIs with immunotherapy. So some patients will, wonder if they can combine these drugs, like add Keytruda to their ROS1 inhibitor. And we generally don't do that because there's a long history of those types of combinations leading to pretty significant side effects or toxicities that aren't seen with either drug on their own. But once you put those two drugs together, you now have a kind of unacceptably high rate of side effects. And so we generally actually try to avoid combining TKIs, including ROS1 TKIs, with immunotherapy. And so that means if I'm going to consider an immunotherapy for one of my ROS1 patients, I generally have to stop their ROS1 inhibitor. And that's obviously a difficult decision to make.
There have been several questions. If people are on another TKI and they're stable, it's controlling their cancer, would you recommend that they switch to zidesamtinib?
Mm. Yes, that is also an excellent question. And as you can imagine, it's kind of it's sort of a case by case that we kind of address questions like that. Zidesamtinib is still a pretty new drug, and certainly it's not approved, right? Right. It's hopefully, as you said, Janet, going to get approved possibly later this year. But we don't have the full sort of length of follow-up, so I don't know that we can say yes, immediately switch. I think the data are very, very promising, and I think for resistance mutations and brain metastases, I think if those are issues in patients that those could be reasons why someone might think about making a switch earlier. But if I do have a patient who's been on, for example, crizotinib, our first ROS1 inhibitor for 10 plus years, and I have a number of those patients, as I mentioned, I'm not going to make a switch of their ROS1 targeted therapy unless they're now encountering some, toxicity that they can no longer tolerate. Because I feel like if you've already demonstrated you're a long-term responder to whichever ROS1 inhibitor you're on, I usually don't want to change that.
So, if somebody has been stable for a long time and now is developing brain mets, would you recommend that they do targeted therapy and switch to a new TKI, or would you have them stay on the same drug and radiate the brain mets?
Um, yeah. So again, that's one that depends. I think if often patient preference plays a big role here, Janet, which is why I'm saying it depends. But I would say in cases where it's one small little brain metastases that's shown up, it's 2, 3 millimeters on a brain MRI, patient's otherwise feeling well, we might choose to treat that lesion with radiation and monitor the brain, of course, very closely and maintain that targeted therapy. Um, but I would say more and more what I've been seeing, especially for ALK as well, is that because we have drugs that are so brain penetrant, if a patient's on a ROS1 targeted therapy that doesn't have great brain penetration, so crizotinib for sure, even entrectinib, and they now start to have brain metastases, I have a lower threshold now just to switch them because otherwise I worry we're just constantly chasing the brain metastases and hoping they don't get ahead of us. So I would say in particular on brain impenetrant or less penetrant ROS1 TKIs, if someone starts to develop brain metastases, even a single one, I generally have been having that discussion about switching to a more CNS penetrant ROS1 TKI instead of doing radiation.
Thank you. We've had a couple questions about zidesamtinib effectiveness in certain situations. One is, do we have any evidence that it's effective in pleural effusions? And the other is in bone metastases.
Um, I guess I would say for pleural effusions, it's kind of regardless of the ROS1 TKI, pleural effusions can be difficult to manage. Pleural effusions can over time, while they may easily have been drained initially, over time, the pleural space can become kind of scarred and form these pockets, and it can become difficult to then drain the fluid effectively. And this is regardless of the ROS1 TKI that you may be on. But I would say in general, ROS zidesamtinib has activity. It's not like they lose activity in the setting of pleural disease. There's certainly activity that we've seen in improving pleural disease, but the pleural effusions themselves can present sort of clinical challenges. For bone metastases, I've seen wonderful activity with zidesamtinib, just as I do in other sites of disease. So I don't think there's any specific reason why zidesamtinib would not work in those locations.
All right. If we have somebody who is on a TKI and doing well, but they're having trouble tolerating it, would you go with dose reduction or would you consider switching TKIs?
Um, so again, depends on the TKI. If
Yes.
it's, for example, an early generation ROS1 TKI and they're really struggling with side effects, I guess I would say, well, between the safety but also the efficacy advantage, like why wouldn't we want to switch to a better one, assuming we can access it? I think if you're already on a very good TKI, especially let's say you're already on, I don't know, taletrectinib, and that's a very effective TKI. Certainly, yes, I would consider dose reductions. We, of course, know how to use dose reductions based on the clinical trial data and our own experience using these drugs. And so we do consider dose reductions. What I've seen for repotrectinib, though, is that patients who struggle with repotrectinib due to the dizziness, that imbalance, um, even with dose reduction, they can still struggle with that dizziness. And so I don't know, again, it's worth a try just because we always like to get as much time as we can off of every TKI. But at some point, you may need to make the switch. And zidesamtinib is significantly better in terms of the safety.
Okay. Um, there's a question as to whether we have any data for zidesamtinib being effective against leptomeningeal disease. Might be early for that.
I think it's early to say, but I am encouraged by the responses that we have seen in CNS metastases. And as I showed in my presentation, the preclinical data is very promising, including the sort of penetrance into the CNS. So,
Okay. We have a little bit of time. Could you give us a very quick overview of what the process is for getting a drug approved once it's gone through clinical trials and has data?
um, what the process is? Well, so the process would be that, of course, the most important thing is it's the preclinical data that which then provided the basis for the clinical data. In this case, it's the phase 1 and the phase 2 data that I mentioned. Usually, we have to have, I would say, at least several hundred patients worth of data because we want to have a sort of robust safety data set as well as confidence in the efficacy. The sponsor, and in this case, Nuvalent, has all of that data that they've been collecting on patients who enrolled in the ARROS-1 study. They put together what we call a package that summarizes the preclinical data, especially preclinical tox, and then, of course, all of the clinical data. And that includes a lot more data than what I showed today. So well beyond just efficacy and safety, but also the PK data, meaning that the drug levels that are exposed and that are achieved in patients, so drug exposures, how that drug exposure might correlate with the responses or with safety. So there's a huge amount of work that goes on to assemble all of the preclinical and available clinical data into this package that then gets submitted to the FDA for review. And the FDA works closely with the sponsor to review the data, and they set usually, a date for when their review will be complete and a decision issued. And Jenna, as you said it, we hope it'll be in September.
Okay. A more general question about clinical trials. If a patient has one type of cancer and then has a recent one or develops a second one, which we've had a few people in the group develop breast cancer after having their lung cancer, does that disqualify them from being in a clinical trial?
So I've had this situation as well. And I would say that many trials do unfortunately have an exclusion around patients who have an active second malignancy. So there are exceptions, though, if you have a very early stage, completely resected in situ cancer. Oftentimes they will list that out in the trial eligibility. But in general, if you have a new diagnosis of cancer that requires active treatment, then it will generally make it very difficult to participate in clinical trials.
Okay. I don't want to run out of time to ask this question. Do you think we are getting any closer to a cure of stage 4 ROS1 positive cancer?
Well, that's why I ended with my final slide because I do. And I think that's incredible. Because when I think back to 20 plus years ago when I was first training, we would never, ever have thought that would be possible. But I think in the setting of specific types of cancer like ROS1, like ALK, and others, where we really understand the biology of those cancers and where we have these incredibly effective drugs that and we've seen the development and optimization of these drugs. I do believe that we're getting closer and closer to a cure, but I think that these targeted therapies, as good as they work, I think they by themselves generally are not going to be able to cure most patients who have advanced disease. And I think what I pointed out in that last slide is that we probably will need to bring in other types of therapies of, we often say orthogonal modalities, so therapies that work in a different way than these targeted therapies. So you can try and basically attack the cancer in 2 or 3 different ways. So, for example, instead of just using the ROS1 targeted therapy by itself, we might need to use the ROS1 targeted therapy with something like chemotherapy or another type of targeted therapy to further try to cytoreduce and eliminate as many of the cancer cells as possible. And then I think a very exciting approach that I mentioned that all of us, I think, in the space of oncogene-driven lung cancers really wants to pursue is how do we eliminate those residual cancer cells that somehow are able to survive despite seeing a really effective targeted therapy. And so there are lots of strategies that we have been already doing and studying now to try and eliminate residual disease. And we really believe that residual disease is really the nidus for the eventual emergence of resistance. So if we could eliminate residual disease, perhaps we could then prevent resistance. And so I mentioned the possibility of bringing in radiation to residual sites of disease, even resecting sites of residual disease. And there may be other medical approaches that we can take as well to sort of target at the biologic level these cells that are able to persist. And so, Janet, I would say yes. I think that we have increasing numbers of tools. And I think the anchor, as I said, is really these incredible targeted therapies, but there's a lot more that we can do to build these multimodal regimens that I think really will offer that possibility of a cure.
That's really exciting. I would love to get us to that point.
Me too.
So, do we have any indication for ROS1 positive cancer whether the effectiveness of these drugs is impacted by age or gender or other aspects that we could take advantage of?
Hmm. Um, no, and in all the analyses I've seen, I haven't seen that. I haven't seen those factors really impacting the effectiveness of targeted therapies oftentimes. But it's a good question, right, Janet? It's getting to how can we identify those patients who are going to be the really long-term responders to a targeted therapy versus those patients who may not have as long-term of a response. If we could identify those factors, that would be really helpful, right? And I would say we don't know. I don't know of any clinical features that do that. We are, of course, looking at molecular features that could potentially predict, say, shorter outcomes or shorter durations of response with targeted therapies.
All right. Well, we have just about a minute left. Do you have any closing statements you'd like to share with us?
Well, thank you so much for having me. It's always a pleasure to talk with the ROS1 community. I'm really excited about how far this field has come. Again, over 15 years, we've come so far, and a lot of that is because of the work that our patients, Janet, you leading the Ross Wonders have really helped us with, right, in terms of participating in our trials, helping to fund research. And so thank you for that. It really has made a huge difference. I think in the research that we can do, the clinical trials that we can do, the questions we can ask, and it really does bring us closer and closer to these incredibly effective therapies. And I hope one day a real cure for patients who have advanced ROS1 positive lung cancer. So thank you.
Thank you for participating with us and for partnering with us in moving forward on our research and treatments.
Thank
And
you.
we hope to have you back.
Thank you. Have a great evening.
Thank you.
Take care. Bye bye.
Bye bye.